Hi, thank you very much, Elisa, for agreeing to give a Build a Cell seminar, and you can get started.
All right, thank you. Thank you so much for the invitation. Our group works at the intersection of, engineering and molecular biology, in particular, DNA and RNA nanotechnology. So today I will talk about a recent project. These are all results that are pre-printed but not published yet. And I'm going to give you a kind of a high-level overview of what we did on trying to build organelles using RNA, why would we do that? So the idea of building organelles is rooted in the interest toward understanding, how does life take advantage of separation? Life is essentially based on separation. To define an individual cell, you need a way to separate it from the rest. And even within a cell, you have organelles, each having its own function, and they have to isolate reaction, recruit components to perform those reactions. And sometimes it is necessary to split them into different compartments. So in biology, the paradigm for understanding separation has been through the presence of lipid membranes, because, if you look at cells, they have a membrane. And many of the organelles inside the cell also have a membrane. But there have been so many discoveries in the last decade or so that have let us identify the existence of organelles that do not have a membrane. In fact, they're called membraneless organelles. And the way they assemble, the way they form, is through the physical principle of phase separation. These organelles are also called condensates because the word should make you think of the fact that there is a phase transition, between molecules that are dispersed. Imagine molecules of water in the air at a certain temperature. But if you cool down the environment, water condenses, and so can many other types of molecules, in particular biological molecules. They can do that. So in 2009, a seminal paper by Cliff Brangwyn and co-workers demonstrated that there are, certain organelles inside embryos. So you see the video here. I hope you can see my cursor, the video on the top left. This is a C. elegans embryo. And those green dots are P granules. And they figured out a way to label these organelles and monitor how they evolve over time. You can see that they appear, disappear. Some of them fuse, some of them shrink and grow, and they eventually migrate toward one of the poles of the developing embryo. So this was kind of one of the first demonstrations that these organelles are indeed behaving like a liquid, and there was the parallel drawn to the existence of phase-separated organelles. Later on, also the group of Cliff Brangwyn demonstrated that some organelles, membraneless organelles like the nucleolus, and these would be the pictures at the bottom, they are liquid, but they can also maintain a structure. So they look like a Russian doll architecture of different phases that are distinct in their composition. And of course, there's been an explosion of studies since the early papers by Brangwyn studying phase-separated organelles. So people have identified a variety of distinct organelles. They have different composition, which defines their identity, different physical properties. So some of them are more liquid, some of them are more solid-like, but they also have been associated with very different functions. Although, one could say overall these organelles can be associated to, gene regulation, stress response, and certain diseases, in particular neurodegenerative diseases. So, of course, our work is motivated by all these exciting discoveries. We want to understand, in particular, how to make organelles from scratch, artificial ones, because the ones that I just discussed, there's so many of them and they're so complex. They include thousands of RNA and proteins. So we want to understand, from the bottom up, how would you make an organelle like this? And of course, we're motivated by all these observations of the biological importance of organelles. But our interest is also motivated by, intriguing hypothesis one could make about how condensate could be associated with the origin of life. So more than 100 years ago, Alexander Oparin proposed that the first, biological entities that one could describe as cells could have actually emerged because of interactions between proteins, small molecules, nucleic acids that could have, created the first separated objects with their own metabolism, and they might be a key step in the theory, for the origin of life. So there's many different reasons why working on condensates nowadays is very exciting. So as you can imagine, the increasing interest in condensation has spurred research toward, okay, how do we make condensates from scratch? And in fact, there's many beautiful papers that came out in the last 5-6 years that focus on how to make organelles, condensed organelles through proteins, typically engineered proteins that harvest motifs from natural proteins that are known to undergo phase separation. Usually they include intrinsically disordered domains and RNA binding domains that will promote non-specific interactions between the proteins and between proteins and RNA that can yield this emergence of dense droplets. Then there's also efforts from the perspective of synthetic polymers or lipids. There's many ways to engineer these non-specific interactions with exciting results in making multi-phase droplets there's a very rich landscape in there as well. And then people have also started thinking about, well, how could we build DNA and RNA molecules that achieve condensation? And this is the part that I will focus on in my talk. I will go back to this idea of, building motifs that can, interact toward making phase-separated droplets. Now, in particular, my talk will focus on RNA. And, why RNA? Because RNA is basically found in all forms of life, from viruses to mammalian cells, which means if you were to identify motifs that can give you phase-separated RNA condensates, they could theoretically be ported to a variety of organisms with appropriate tweaks, but they could function. RNA is a molecule that in cells has many functions. It stores information, but it can also have structural functions. This is a video of like a rendering of ribosomal parts, right? So ribosome is a structural component and it's largely made of RNA, but it also, of course, aids in the processing of information. And another great advantage of RNA as DNA, of course, is that we have a good models for predicting structure starting from sequence and vice versa, right? So if we want a certain structure, there's software that allows you to optimize the sequence so that you can achieve that folding. So these are all great properties of RNA, and it's still much easier to engineer RNA than it is to engineer proteins. Now, there's other groups, of course, working on developing RNA condensates. So in fact, the first attempt to do that, I think, was by the group of Ron Bale. So in this paper from 2017, they demonstrated the formation of RNA organelles inside mammalian cells using very long sequences of RNA that include triplet repeats. So many repeats of sequences like CAG could give you, the emergence of these RNA-dense regions. More recently, a similar approach has been used to develop condensates inside bacteria. And there's also results in this direction from using multimerizing aptamers. So essentially cloning many repeats of aptamers like beetroot and broccoli. They could give you they can cause RNA interactions that then form bright organelles, as you can see them here. These are labeled with fluorogenic aptamers. That's why we can see them inside mammalian cells. So the model for these previous attempts to make RNA condensates, successful attempts and very exciting results, is that of essentially creating a molecule of RNA that has a certain number of interacting bonds that here I am depicting with these orange dots, okay? And these very long molecules, once they begin interacting, they sort of form a tangled mesh where the individual bonds between the molecules, because these repeats are sufficiently short, these interactions are sufficiently weak, you have constant binding and unbinding of these domains. And so that's how you maintain a liquid or gel-like state. But fundamentally, all these molecules make a big mesh, a mesh that is disordered in some sense. So the problem that we saw with these approaches is that it's hard to scale these architectures in terms of sequence design, okay? You know, it would, there was no, in our opinion, there was no clear strategy that you could follow to now take this idea to make and scale it to make many distinct organelles. And of course, because they are tangled complexes of many long molecules, it becomes a bit hard to control their properties, okay? So we went back to DNA nanotechnology and the lessons we learned from that to figure out how to make RNA condensates through a different approach. And the idea we started from was this idea of using star-shaped motifs. These were demonstrated using DNA molecules already more like a decade ago by Biffy, Shortino, and co-workers in 2013. Later on, the group of Omar Saleh at UCSB also started, developed many interesting demonstrations on this, and more recently, the group of Takinue at the University of Tokyo also has several exciting results on these nanostars. So as you can see here, you have essentially these nanostars are built from 3 or 4 distinct DNA sequences. They bind forming this junction, and they have sticky ends at the end of the arms, okay? So you can sort of abstract this motif to be like essentially a star that is scaffolded by these blue domains that are not responsible for intermolecular interactions. But then you have binding sites at the end of the arms, and those are the sites that create nanostar to nanostar bonds. And if they are, and usually they have to be designed to be sufficiently weak, but also sufficiently specific so that you can create distinct nanostars that only interact with their counterpart, okay, with nanostars that are complementary to that. So here you have an example in these images that I already flashed earlier, okay? So here you have like pink nanostars and yellow nanostars. They differ not in their structure. The structure is the same by the differing sequence, okay? The sticky ends of the different sequences. And so they form distinct compartments that are labeled here in pink and yellow, okay? They have different dyes associated to it. But also you can link the motifs through these, darker shaded nanostar that has half of the sticky ends are yellow and half are purple. That indicates distinct sequence. And so depending on the amount of linker, you can create condensates that now are perfectly demixed, or they are like, a mix of yellow and purple. So the idea is that this motif allows you to control very clearly the valency, so the number of bonds you have, the avidity, the strength overall of the bonds, and also the size of the molecule, because you can tune the length of the arms so that the general volume occupied by a nanostar is clear. And you can actually change the number of arms. So that's how you control the valency. As you can imagine, you could have a nanostar with 3 arms, but also with 6. They're modular. In fact, you can change and replace these sticky ends and make sure, and they have an ample sequence design space. You can program their phase diagram. There's papers that have clearly demonstrated you can do that. Program how they transition from liquid to from gas to liquid, and you can change. You can control the temperature at which that happens depending on the sticky end length. And they also have tunable kinetics of formation. So this is kind of where we started from. This is what we have learned from DNA nanotechnology. How do we translate this to RNA? And so the first attempt, of course, we took this architecture and we simply, re-optimized the sequences so that we could build this nanostar using RNA. And this is work that was actually led by Dr. Jamie Stewart. She's now a professor here at UCLA, and Paul Rothemund at Caltech. And here's the preprint if you want to check it out. So with these nanostars, remember the sticky ends are self-complementary. So you can, design the sticky ends so you get either dispersed, phase, droplets, or gels, depending on the interaction strength of the sticky end. So these three images show you results from nanostars that have a different length of sticky ends. But you can also tune the strength of the interactions by changing temperature and the ionic conditions. So these condensates include, I believe, a fluorescent dye, but you could also append fluorogenic aptamers to the nanostars, and you can get like macroscopic gels that can be they're like, this is clearly like a condensed RNA mesh. And you can also recruit to these nanostars some labeled peptides, so you can separate peptides in different compartments. But this is not the main topic of my talk. I just wanted to give you a sense of the fact that you can take the DNA nanostar architecture, translate it to RNA, and it will work. Now, what my group has worked on is a single-stranded version of these nanostars. And here you can see immediately one difference, that we have 3 arms instead of 4, because this way we get a more compact structure. And now this is a single RNA molecule that folds into a sequence of stem loops, where the loops that are marked here in red, they are chosen to have kissing loop sequences. There are some unpaired spacers in the middle of the junction, so to give more mobility to the arms. And of course, there is a nick here in the middle because it's where transcription begins and ends. So you can abstract this molecule with sort of like a 3-stick figure, where the red dots are where the molecules bind to each other. They're self-complementary, they're palindromic. And again, if you design the sequences to be strong enough, but not too strong, you can get the formation of these mesh of small interacting RNA molecules that condense, okay? So they interact through kissing loop binding. And here are also like 2 preprints that we have recently shared. The one on top is primarily our work. The bottom is by Fabrini and others is a collaboration that serendipitously happened. Actually, this is an interesting story scientifically, because both groups sort of developed independently these nanostars, and at some point we realized we were working on the same problem, so we decided to work on it together. So it was a really nice experience. So this is the idea. Now, how do we demonstrate it works? So first, we tried to transcribe and purify the RNA. Once we have the RNA, we anneal it. We have first a melting step, 70 degrees, and then we cool it down, and we hold the sample at a particular temperature. We will change it between 40 and 60 degrees. And once we wait long enough, then we watch the sample to see if condensates have emerged. So with this type of protocol, we tested different variants of kissing loops. The kissing loop variants were mainly taken from the paper cited down here. And it was a study on the HIV kissing loop sequences. Was studying what are the requirements for kissing loops to bind, even if you have mutations. And here basically we took the same variants that they considered in that paper. We took an annealing temperature hold of 50 degrees, and some variants produce condensates and some not. And interestingly, the results are quite consistent with what they found out in that paper I referenced to. Kissing loop variants that are predicted to form dimers in this paper are those that generally also gave us condensates forming on these nanostars. Now, these nanostars do not have a label. They're not labeled with an aptamer or with a protein. The way we image them is by adding cyber gold. Okay, so first we have the production of the condensates, and then we add cyber gold at the end, and that's how we see them. So you can do image analysis here. You can use, so because not all of them are spherical, it's actually hard to develop software that will sort of segment droplets, calculate the diameter, and so on, because in many cases, they're actually not spherical. Some of the samples gave us gels. So what we did instead was doing chord length analysis. It's image processing method where you essentially segment your image in horizontal and vertical lines, and you look at the analysis, and you analyze the length of chords of continuous stretches of material. And so the chord length analysis is what I'm plotting here. These are violin plots of the chord length analysis. So here, what you can see is that, of course, at lower temperature, it is more likely that your motifs will condense, as it would be intuitive, I gave you earlier the example of molecules of water. For water to go from a gas phase to a liquid phase, you need to lower the temperature. So similarly, these RNA molecules will more likely give you condense into droplets if you lower the temperature. And there is notably only one variant that didn't give us any condensates, but all the others produced some form of dense RNA mesh. So here we change the kissing loops. What happens when we change the stem? So one thing we noted, and this is the top series of data here, is that the longer the stems, the larger the droplets that we image. And so, of course, this is only one snapshot in time. They hardly reach a stationary distribution of sizes because they are droplets. If they are liquid, they continue to fuse. There's a collision between droplets, as you can see, for example, in this case here that I'm pointing with my cursor, they will fuse, okay? And by fusion, continuous fusion means that at some point you should, at equilibrium, you should have only two compartments, the dense phase and the gas phase, the dispersed phase. But at this particular snapshot in time, which is the same for all three samples relative to the start of the experiment, a longer stem helps you generate bigger droplets, which intuitively makes sense. And we saw the same thing actually with DNA nanostars. So it's a consistent result. But so not only the size of the arms matter, also the sequence matters. And so we took two different stem variants. And as you can see here, the different kissing loop variants that we tested in one, in some cases, they work also in the second stem, and in some cases, they don't, okay? And here, we're still trying to figure out why. It's not clear, it's possibly neighboring interaction near the kissing loop, or perhaps some of the stems are unfolding properly. We don't have here a clear, explanation of why some sequences work and some don't, but beware, if you try to do this, the stem will make a difference. Another thing we tried was to change the sequences of the kissing loops so that they are not palindromic. So here we actually designed arbitrary sequences that have nothing to do with the HIV kissing loop. And we created a 2 nanostar system, Nanostar 1 and Nanostar 2. They have complementary sticky ends, and so you need both nanostars in order to form a condensate. One alone is not enough. And so we have 4 sets of sequences here that worked. And we did verify that, if you take, say, one in set A, you take only one of them and you purify the RNA and anneal, it won't form condensates, okay? So in a way, this gave us, this confirms that the interactions are actually mediated by the kissing loops and not actually by misfolding of the stems during annealing and stem-stem interactions. I think we can exclude that. So next, we thought, well, in nature, condensates are, useful to recruit stuff, to isolate reactions and molecules. Can we do the same with these RNA condensates? And the answer is, of course. So proof of principle, we tried to partition small molecules, in this case, DFHO, okay? And we decorated our nanostars with aptamers for red broccoli and for corn. We tried also other things, but this is one example that worked particularly well. So each aptamer is now recruiting DFHO, but as it binds to its own aptamer, DFHO emits light in a different spectrum. So you can see the yellow and the red condensates here. The two nanostars have different kissing loops, so they don't interact with each other. That's how you can see some that are yellow and some that are red, and there's no orange, okay? So we did a systematic image analysis where we actually looked at the pixel intensity. For each pixel, you can calculate an intensity in the plane FITC Psi 3, which is the two emission spectra. And we looked at the angle, okay, of that particular point. And we plot here, you can see this is a histogram of the angle of the pixel intensity. And you can run two experiments, one where you have separately the red and the yellow, and that's the bottom, that's our control, okay? So you can see they have distinct peaks. And then you can compare that to what happens when they're simultaneously annealed together. And the pixel intensity spectrum is consistent, and it has like two main peaks, okay? So that tells us that it's not just this image that we're looking at that looks particularly nice, but it's a general property of the whole sample. So now enough with this. So as I said, this was our first attempt to characterize these condensates. We purified the RNA, annealed, and then washed the sample. Now, next, of course, we wanted to verify if these condensates could form during transcription, which they should. That's the reason why we came up with this design with a single strand, because RNA folding, in general, should take milliseconds to happen, which is a much shorter time scale than RNA transcription. So as soon as the RNA is generated by T7, it should already fold correctly and form this sequence of stems. And stem dissociation likelihood is negligible. It's very unlikely that the stems will come apart unless there is, a remodeling event caused by other molecules that are complementary to that. But that's first, we need to verify that this works, right? So the next set of experiments are what we call co-transcriptional experiments. So we add the DNA to a sample where you can have that has the right reagents to do in vitro transcription. We let it sit at 37 degrees for a few hours, and then we add CyberGold to visualize whether condensates have emerged or not. And so they do form co-transcriptionally, which is great. Okay, so the top row shows you an example series of images for a single nanostar case. So here you have the HIV kissing loop palindromic sequences, or a variant of that. I don't remember now which particular sample this is. And here you can see the chord length analysis. After 1 hour, you can already observe, structures that have on average a 1 micron length. Okay, and at 4 hours, you can see that it's hard to distinguish now individual compartments, and they seem to fuse quite slowly, which tells us this is probably more closer to a gel than a liquid. And then we also tried the 2 nanostar case. So here we have 2 distinct genes. Each is transcribing its own nanostar, but the 2 nanostars have complementary kissing loops. And so already after an hour, we have comparable lengths, but qualitatively the sample, again, looks more like a gel than a liquid. So that tells us that if we wanted to get spherical droplets here, we would have to do a little bit of work in sequence design. But this is, again, so only at this stage is proof of principle that we can obtain this type of architecture. So next, these reactions were done in high-yield transcription buffer. There's nothing else there. But we wanted to see if this works also in TXTL TXTL samples. And we use Pure Express, which I know a lot of people working in cell-free systems are adopting to make protein, okay? And here, all we had to do was add our DNA template. It has, of course, a T7 promoter, so it can be transcribed by the standard components of Pure Express. And here we observed similar, kinetics of growth as what we saw in the transcription buffer, although actually took a little longer for the condensates to form. But Pure Express, I don't think it's optimized to have high yield transcription, and it has a lot more stuff. But this is interesting because it's telling you, if anybody wanted to try these condensates together with protein translation, say you want to make a protein and see if it gets recruited to the condensates, you could try that because they work, okay? This is really taken Pure Express, taken out of the box, no optimization. And in fact, that's sort of next what I'm going to show you is that you can recruit peptides during transcription to these condensates. So all you have to do is have an aptamer for your favorite peptide, and to verify whether this works or not, your peptide should be labeled with something, okay? And as a proof of principle here, we took peptides for BoxP and TAT, which are often used as aptamers to then recruit proteins that are fused to the BoxP and the TAT protein. And so, here, this data tells you that appending the aptamer in the absence of peptide gives you condensates with a chord length distribution that is comparable to what we saw before. But interestingly, the more aptamer you have, okay, the larger the condensates, the larger the chord length distribution, the wider it is, which makes sense because in a way you're making this nanostar a bit bigger by adding an extra arm. The other arms are more likely to stay more stretched. So then we co-transcribed the RNA, and then we spiked in the peptide. The peptide here is not translated in the same sample; it's just added. And we could we saw that the BoxP aptamer recruits specifically its own the P22 peptide, but not the TAT. And vice versa. Okay, so it works as intended, and it has enough specificity, at least for these two aptamers that we tried. Of course, there's an infinite amount of aptamers that one could pick, and so it would be fun to see if this result holds for other types of peptides or proteins. And I want to mention this work was done by Martin Gobri. He was a visiting student in my group this spring. And in really only 3 months, he was able to get these nanostars to work under his hands, and he got these interesting results with the peptides. So I'm almost finished. So the last thing I wanted to mention is that, of course, now we're interested in seeing if these condensates can be made inside cells. So we spent quite a bit of time cloning the nanostar design into a plasmid, transfecting it into cells, and then seeing if it can actually work. And this is, preliminary results, but they're very exciting because indeed we saw that we could make RNA organelles inside different types of mammalian cells. So the data I'm showing you here is for HEC, but we have them working also in HeLa and in osteosarcoma cells. The reason these condensates are fluorescent is in this case, we added a broccoli aptamer, and so we can visualize them easily. So negative controls, of course, did not show condensates. And another negative control where we simply express broccoli aptamer without the nanostar. So these show like diffused green fluorescent in the mostly in the cytoplasm. And note that there are some dots here in the negative control, but we have found that this is known that broccoli can co-localize in specific cellular organelles and create dots of this kind. But this is, as you can see, qualitatively very different from the organelles that are generated by the nanostars. Interestingly, we saw condensates both in the nucleus and outside. And there are many features that we're trying to investigate about this system. So this is all work in progress. Our goal next is to figure out how to localize them to specific subcellular locations, whether we can recruit pathways to the condensates, and also potentially sense events that are happening in the cell by generating the organelles. So that concludes my talk. So I hope I gave you like good understanding of motivation. Why are we doing this? We feel it's a very exciting area of how to build synthetic organelles using condensing molecules. I showed primarily results in vitro, okay, both in transcription buffer and in pure express. And the perspective now for us is that of working inside cells to make artificial organelles. And with that, I also want to thank the students and postdocs in my group, the group of Paul Rodemund, and then Catherine Platt and Douglas Black here at UCLA. They're biologists who have been advising us on the next steps for the in vivo work. And of course, the funding agencies that make it possible for us to do the experiment. And with that, I will take any questions.
Thank you so much. That was a fantastic talk. There are several questions in chat. If you could read a question out loud before answering, that's going to be great for a recording.
Okay, so we have a question. Could you please comment on temperature compatibility with cell-free protein or using synthetic cells? Okay, so I think that question was answered during my talk because you can make these things co-transcriptionally. Our initial characterization was done through annealing because that's the gold standard when you characterize DNA or RNA nanostructures, to do annealing to get rid of secondary structure that is potentially unwanted and make sure you reach the thermodynamic equilibrium of the structure. But because of the way they're designed, because these stems form immediately during transcription and they're very stable, you can run this at 37 degrees, no problem, no need for anneal. Okay, so then there's another question that I think demonstrating synthetic condensate in the cell would lead to interesting applications. Could you give us the relevant challenges to do so? Okay, so yes, there are. So it took us quite a bit to be able to clone the nanostars in a plasmid. The reason is these stems don't clone well. In fact, we tried to get this onboarded on Twist, and they won't do it. So it's a bit laborious. Someone needs to manually clone and figure out the right way to clone. We're not ready to, of course, distribute details yet because we're not close to publishing this at this point. But it wasn't easy to. I think that cloning was the hardest part. And then you need to figure out, mammalian cell, you need to figure out the right promoters to have enough expression of RNA. Those are the main two challenges. So, okay, another question. Do you think?
If I allowed to ask additional questions. So cloning and promoter and everything also very challenging, I know, because recently I also started the mammalian synthetic biology. That's not fun at all. But at the same time, I just wonder, environment may have some impact how those condensates behave, because in the in vitro system or cell-free system, you have everything under control, but in the cell that might be different scenario. Of course, I mean there are some natural condensate, but this is a synthetic system. You will have the difficulty in terms of the ratio of these partners and so on. At the same time, the environment is already different because you have the other compartment as well as other organelle in the cell.
Yeah, of course, yeah.
So I just wonder, how they potentially impact, successful demonstration in the cell. That was actually my question.
Right. Okay. So I think it's too early for us to answer that in the sense that we're still at the early stages of characterization. We did notice that some cell lines did not like to have the condensates expressed in them. Some others, no problem. Like the image I just showed you in hex cells, this was our first try. Like it just worked right away, which was remarkable. We weren't expecting it. And the condensates seem to appear very early on after transfection. But to give you a more precise answer, I mean, of course, we would have to genetically encode the sequences somewhere in the genome and then make it inducible. That's what we want to do. But as you can imagine, it's not something you do overnight, and we're working on that.
So yeah, I see the huge potential once you successfully demonstrate that one, because there are lots of applications you could imagine. So
Yeah.
I'm looking forward to you achieving that goal. I mean, in near future.
Thank you. Yeah,
Yeah,
we're very, very excited. Right now, this is kind of taking all the energy of my group. We're all thinking hard about it. Okay, so then let's see. Do you think it will be possible to solve the structure of the phase-separated RNA droplets by cryo-EM? It's a great question. I don't know. I'm not an expert in cryo-EM. I think it would be awesome to do it. We don't have the capability at the moment to do it. So if you know anyone who's interested, send them my way. It would be lovely. So what is the program you use to predict the folding of nanostars? NUPAC. That's the software you use. Can one of the terminal ends be extended to use protein with single-stranded binding domains instead of aptamer for protein recruitment? In vitro, yes, I think so. We haven't tried. We have tried to extend the 3' or 5' end of the nanostars to recruit for other purposes, but it does recruit other oligonucleotides. For proteins, we have not tried. No, that's not what you're asking. You're asking single-stranded RNA binding domains. Yes, you can do that. Of course, you have to pay attention to which sequence you include, because if it causes misfolding of the nanostar, then it won't work. Then what sets the size of these nanostars? Do they reach a critical size? So that's a great question. They keep growing, because if they're liquid, as soon as two droplets collide, they're going to fuse. So, and this is a problem with any kind of condensate, not just RNA. It's hard to maintain size. They form, and then by virtue of the mechanism that they form with, they will continue to grow. So imagine you're making a vinaigrette at home, right, with oil and water. You can create an emulsion, but then if you wait long enough, eventually the oil and the water will just become separated. The oil will float on top, and the water will be at the bottom. So that's the same thing with any kind of RNA condensate or any kind of biological condensate that is liquid. But cells in some way maintain condensate size, because organelles don't grow unbounded, right? So we're thinking about that from a theoretical perspective, and we're starting also to identify some experiments that we could run to introduce some sort of chemical reaction that goes on between the dense phase and the dispersed phase. That there must be some flow of material through chemical reactions that allows you to maintain a size. But at the moment, no, they don't reach a critical size. In theory, they could grow without end. The next question, if you comment on the emergence of condensates using pure express, what kind of interaction happens while protein is being expressed? So when it's okay to image that lysate. We tried FRAP. Okay, so one thing at a time. So we did not do protein translation simultaneously as condensate formation. So we don't know whether if you were to say express GFP and try to then recruit GFP to the condensate, what would be the interplay between those two processes? We don't know. But because they form in pure express, you could do that. So that I think that would be fun to try. So is it okay to imagine that lysate component and tRNA from pure express contributing to increasing the size of condensate by sequestration? It's possible, actually. That's a good question. We didn't try. We didn't ask. We didn't try to quantify that, but it's possible that tRNA could be incorporated. If there is any complementarity, I think it could. In the absence of complementarity, I don't think so, because RNA backbone is negatively charged. So if anything, these condensates should exclude other RNA. They shouldn't attract it spontaneously, just based on charge. But of course, it depends on the environment in which they're in, because if there's lots of positive cations, they could create screening effects that will limit that negative charge repulsion. But that's something that needs to be studied. Have you tried FRAP on condensates to see the diffusion coefficient? So there's no proteins in our condensates. They're pure RNA. There's no proteins at all. But we don't have yet FRAP data. We are in the process of doing that. But that's a great question. So a question. Hi, Michael. You're here. It's exciting to follow for applications in cells. Can you comment on how the nanostars are affected by cellular context, such as RNases or having terminators extended on the nanostars? We haven't tried that yet. We haven't tried to add domains that would limit RNA's influence or terminators. We just transcribed the nanostar design as is, and we saw the condensates, so we're happy with it for now. Related to that, is there any consideration on whether the double-strandedness kicks off the RNAi pathway? Great point. I don't know. That's definitely something to think about. Yeah, thanks. Great question, but I don't know. And I think that's it. I don't see other questions.
Yeah, I think you went through all of the questions. Some of them were what I would ask myself, so I think I don't have any other ones either. Thank you so much, and thanks everyone for a great discussion.
Yeah, thank you so much for the awesome questions. And if anybody is interested, contact us. We would love to collaborate and get this work, especially the work in vivo, get it off the ground.
And have a great week, everyone.
Thank you. Thank you so much again for the invitation. Bye.
Bye.